This is part 2 of the nucleic acids lecture, and now we're going to be talking about how it is that DNA replicates itself inside of this cell. And just to give you a heads up, the process of DNA replication, or also called DNA synthesis, is an event that happens right before another event called mitosis. Okay, and mitosis is the division of a cell into two daughter cells. We'll learn more about that later on in the semester. But before I can even divide a cell into two daughter cells, I have to make a complete copy of all of the DNA within that cell so that each daughter cell has a complete copy of the DNA. So there are a number of different players that get involved in this process of replicating DNA. Of course, first you have to have DNA present in order to be able to copy it. And you're going to find out that we need something called a primase. Primase is an enzyme. It ends in ASE, so you know it's an enzyme, that is responsible for putting a little primer piece of nucleotides on the DNA to get the process of replication initiated. And we'll discuss that in a minute on the coming slide. You need something called an RNA primer. Okay, that's the little piece that gets put on there. DNA polymerase III is the enzyme that does the bulk of the work in terms of actually copying the DNA to create a new DNA molecule. Then we've got some other players like helicase, which helps to set up replication so that it can occur. You have to have a bunch of adenine, thymine, guanine, and cytosines that are free floating around that can be used to manufacture the growing new DNA chain. And those are called single-stranded DNA binding proteins. I'm sorry, those are called the nucleotides. Sorry, I jumped ahead of myself. You also need to have single-strand DNA binding proteins, and these guys are kind of like little chaperones that sort of assist in the process of replication. You need something called topoisomerase I'm going to talk about in a couple slides. And lastly, you need some glue to stick everything together, and that glue comes in the form of something called ligase. So let's take a look at this process and the steps in it. One thing I really want to stress, it's very critical that you do, go online to our online classroom site and be sure to view the animations I posted there, because frankly they do a really, really good job of explaining this event of DNA replication. I can certainly explain it, but it's nice to be able to see the animation because you can see in real time, in real movement, what's actually occurring rather than just trying to imagine it in your head as I explain it. So please, please, please do view those animations. They will help you tremendously in your understanding of this topic. So let's start out and talk about a few things, a few terms that I'm going to be using in the next slide, and I want to make sure that everybody knows what those terms mean. So let's get everybody on the same page. One thing I want to remind you all again is that DNA is a directional molecule. It has a 5' end and it has a 3' end. And you will remember that the 5' end is the end of the molecule that ends in a phosphate. The 3' end is the end of the molecule that ends in a sugar group. And when you go to watch the animation and when you are listening to me go through this step by step on the next couple of slides, you will notice that DNA replication moves in the direction of 5' to 3' on the new strand. Okay, the new strand of DNA that's being manufactured is always manufactured starting at the new strand's 5' end and moving in the direction towards the new strand's 3' end. Very important. Another pair of terms you're going to hear me talk about are leading and lagging strands. One strand in the DNA replication process is able to manufacture a continuous string of nucleotides. That's called the leading strand. We will see that the other side of the DNA molecule that's also being copied is being copied in little chunks or fragments, and that is called the lagging strand. We'll see why that is shortly. Remember again, as I told you before, we have to have primers to get this started. Just like you have to prime a gasoline engine in your car to get it to start, or just like you have to prime a lawnmower to get it to start, we have to have primers that help to get the process of DNA replication started. And those primers are RNA segments, okay, little segments of RNA. And lastly, you're going to hear me use a term called Okazaki fragments, and this relates to what happens on the lagging strand. And remember, I said that on the lagging strand, we are making the DNA in little short chunks. Those little short chunks are called Okazaki fragments, and we will get to that shortly. Now, before I go on to the next slide, I want you to take a look here at this picture down here. Remember that in bacterial cells, the DNA that they have is not found inside of a nucleus. It is free-floating in the cell. And if you didn't know this already, bacterial DNA comes in the form of a giant circle or loop. And when DNA replication occurs, you have these little bubbles that take place on this little loop of DNA that then begin to expand outward and enlarge as we copy the DNA. By comparison, if we look at eukaryotic cells, the chromosomes in eukaryotic cells, such as our cells, are not circular. They are, in fact, long segments that can be up to billions of nucleotides long. And those long segments called chromosomes become bubble-like, if you will. During replication, there will be multiple locations along that chromosome which are opened up in order for replication to begin to occur. And those places where the chromosome is opened up so that copying can take place are called replication bubbles. So I think we're ready to go and let you go ahead and actually view the animation rather than me go into detail, because the animations do such a good job. We will then revisit this quite in depth in class. But instead of me lecturing on it, I think the animations are going to do a better job for you. So keep in mind those key points to watch as you're viewing the animation. Particularly remember that you have to have a primer in the form of RNA to get the process of DNA replication started. When we meet in class, as I said before, we're going to spend quite a bit of time going through this together to make sure you understand the finer points of it once you've at least overviewed it on your own before class. Now, what happens if during the process of DNA replication, what happens if we make a mistake? DNA polymerase III is the main character that does all the copying of DNA inside of a cell, and it has a pretty good, pretty low error rate. Once it goes through and proofreads its own copying, its error rate is about 1 × 10⁻⁷, which is about 1 in a billion errors there will be. Okay, so that's not very many errors. That's actually pretty good. But imagine if one of those errors that just happens to slip through happens to occur inside of a gene that's key for you being able to function properly. This is what happens, for example, in people who have sickle cell anemia. It's a change of just one little error or mutation in the DNA that creates a pretty profound disease for these individuals. Anytime that you have a copying error, that is called a mutation. And these happen spontaneously. Every time your cell copies itself, it is going to have at least a handful of mutations that occur. Now, fortunately, most of the DNA that you have does not contain genes. Only about 3 to 5% of all of your DNA actually has genes in it. The rest of it is DNA sections that may support manufacture of proteins or may have other duties, but they themselves are not genes. So usually when we get a mutation, it occurs in those regions that we call non-coding regions, those regions of the DNA that do not have a gene present in them. All the same, sometimes you can get mutations in genes, and as I said before, it can have a profound effect. Now, as I said, DNA polymerase does proofread what it does in terms of copying DNA, and it does have a really low error rate after proofreading is done. If it does find an error, to correct that error, it can use a process called mismatch repair. And what happens here, if we look at this picture, let's say that this section of DNA, you can see here a T is being paired with a G, which we know is not correct. T should pair with A and G should pair with C. So what happens is the DNA polymerase will see that mismatch and actually will have DNA polymerase 1 come in and chop out the segment that surrounds where that error was. So DNA polymerase 1 comes in and chops out the error region, and then DNA polymerase 1 also then fills in the area with the correct sequence to make the repair, and that is called mismatch repair. Okay. One question that puzzled researchers for a long time was, when DNA polymerase 3 is going through to do its proofreading, how does it know which strand was the parent strand? In other words, how does it know which strand was the original copy that's correct, and which strand is the daughter strand or the new strand that could contain the error? That's really key, because if you are going to make a correction, you need to know which side of the DNA strand is the original copy that you have to make the correction against. Okay? And the way that we do that is through a process called methylation. Early on in the replication process of DNA, methyl groups are added. You can see them here. Methyl groups are anytime you have a carbon with 3 hydrogens attached. That's a methyl group. And throughout the parent strand of DNA, around the time of replication, these methyl groups are added onto the parent strand, and that tags the parent strand as being the original copy. That way, when DNA polymerase goes through, if it reads an error, it knows which strand is the daughter strand, i.e. the one that contains the error, and which strand is the parent strand, the one that is the original correct copy, so that it doesn't make the correction in the wrong side of the DNA. Very interesting process. Next up, I want to talk very briefly about another way that DNA can have a mutation occur and how it might be corrected. In this case, ultraviolet light can serve as a damaging agent to DNA. And we all know ultraviolet light is present in the sun. Every time we go outside and get a sunburn or a suntan, that is a indirect effect of ultraviolet light exposure. And if you go on to anatomy and physiology, you will learn specifically why it is you get a tan or a brownish hue to your skin when you're out in the sun quite a bit. But for now, let's just talk about the effect that ultraviolet radiation can have on the body. Specifically, ultraviolet radiation can be quite damaging to DNA molecules at a particular place in the DNA molecules. Any time on DNA that I have 2 thymine molecules close to each other. So for example, let me just draw this out. Let's say this is my section of DNA. You can see here that I have 2 thymine molecules side by side on one side of the double strand of DNA, and they are paired across from them with adenine, as they should be. When DNA is hit or struck by ultraviolet radiation, it has a very peculiar effect on these areas where I have 2 thymines side by side. What happens when I have these 2 thymines side by side is when ultraviolet light strikes them, that breaks apart the hydrogen bonding that those thymine molecules normally have with adenine. So they stop being bonded here, and instead those 2 thymine molecules bond with each other across this way. Okay, so here we have these 2 thymine molecules bonding with each other right here, instead of being bonded across over here with their adenine partners on the other strand of DNA. And why is this a big deal? Well, the big deal is you'll notice that this physically contorts the phosphate sugar backbone. It actually produces a kink or a bend in the DNA strand where the 2 thymines have joined together. And what that does is it actually inhibits proper functioning of things like DNA polymerase 3 or RNA polymerase or other molecules that might want to interact with that DNA to copy it at some time in the future. So how do we repair this? Well, if you have good functional repair molecules inside of your cells, then you undergo a process of repair called excision repair. And in excision repair, the enzyme that does the fixing comes in and actually cuts that bond between the 2 thymine molecules and rejoins those thymine molecules up with their adenine partners as they should be. What happens if your enzyme that's supposed to do that repair is non-functional. Well, you can get some very serious disorders. And a good example of this is a disease called xeroderma pigmentosum. And in xeroderma pigmentosum, these individuals do not have the ability to repair thymine dimers that occur. So anytime they are exposed to sun, they cannot make a repair. And as a result, they accumulate these thymine dimers in their DNA very rapidly because they're not fixed. And as a result, their chances of developing cancer from the presence of these thymine dimers is hugely increased. So if we look down here at this chart, individuals that have a normal amount of the repair enzyme, you'll see their cells aren't very, most of the cells will end up surviving exposure to ultraviolet radiation. By comparison, individuals here in this second one, the second line, are individuals that have xeroderma pigmentosum, and you can see that the number of cells that survive high doses of ultraviolet radiation is far reduced. Another way you can look at this is you can see how many cells actually get repaired after they've been exposed. And you can see in a normal individual, nearly all the cells get repaired, okay, in a normal individual, but almost none of them get repaired in individuals with xeroderma pigmentosum. So that's it for our lecture on nucleic acids. Remember, though, that a big portion of this lecture is you going and viewing those animations. Be sure after you've viewed them, if you've got questions or points of confusion, you write them down and bring them to class so that we can work through them and make sure you're clear in your understanding of how DNA replication occurs. And I'll see you there.